Good morning, everyone. My name is Tremaine Mitchell-Ryan, and I am a scientific program manager at the Health and Environmental Sciences Institute, and it is my absolute pleasure to welcome you to our Immuno-Safety Career Symposium, Spring Break Edition 2024, brought to you by the Health and Environmental Sciences Institute's Immuno-Safety Technical Committee, or the HESI ITC. Today, it is our goal to illuminate the path of careers in immunosafety by leveraging the collective wisdom of our esteemed panelists. So what is HESI? Who is HESI? The Health and Environmental Science Institute, or HESI, is a nonprofit science organization who have for over 30 years now played a pivotal role in resolving global health and environmental challenges. And it achieves this by fostering a collaboration among scientists from various sectors, including academia, government, industry, and NGOs. Today, you will see this collaborative spirit in action as we delve into the world of immunosafety and toxicology. The Immuno-Safety Technical Committee, or the ITC, is at the forefront of addressing issues related to immune safety. Our mission spans from advancing immunosafety science to educating stakeholders about the significance of immunosafety data. And we do this through a variety of objectives that you see listed here below. Also, what is very important to the committee and what we hold very close to our heart is a commitment to sustainability of talent in the field. And this is manifested through various initiatives, including today's Career Symposium, professional development, travel grants, and a mentoring program. These platforms are designed not only to provide insights into the field, but to foster professional growth and development. So here we are today. I'd like to quickly introduce or give you a brief snapshot of the greatness that you will be hearing from today. We are privileged to have with us 4 distinguished panelists from diverse sectors: Dr. Rashadi Haynes from industry, Dr. Denise Wilkins from a contract research laboratory, or Charles Rivers, Dr. Shaji Theodore from government, and Dr. Marie Fortin, who I guess she holds both hats. She sits both in industry and academia as well. Each brings a wealth of knowledge and experience in immunotoxicology and the toxicology field as well. Guiding our fire discussion today is Dr. Nikki B. Marshall and Dr. Marie Soleil-Pichet, who will serve as our moderators. Their expertise and insights will undoubtedly enrich our conversations and help us to explore a multifaceted landscape of immunosafety careers. So I just want to quickly give you a lay of the land and show you quickly show you today's agenda. You've already had my welcome, and I don't want to spend too much more time speaking because I really want you to get to hearing from the experts. We'll have greetings from our moderators, and all of our panelists will provide an introduction of themselves. That will be followed by a brief question and answer session that will be led by our moderators. And then you'll also have the opportunity to pose any questions that you have to our speakers as well. And this will be followed by closing remarks, and then we will adjourn. I would like to remind you that post this event that we will be distributing a short survey to get your feedback on this event, but to also get more information on how we can enhance more programming tailored to graduate students and postdocs. So without further ado, I will hand the podium over to our moderators.
Great. I think I can kick things off. Hope you can hear me okay. Great. So hello, I'm Nikki Marshall. I'm currently a team leader in the non-clinical immunotoxicology and biomarkers group at GSK. Our group handles questions, concerns, testing related to safety as it pertains to the immune system during drug development. And really excited about today's session. We have a great group of panelists. So I'll hand over to Marie Soleil, also moderating.
Hello, everyone. So my name is Marie Soleil-Pichet, and I'm the scientific director of the immunology group at our Charles River facility located in Montreal, Canada. So I work for a contract research organization. I've also been part of ITC for over 10 years now. So as Charmaine mentioned, today I'm going to be moderating this career symposium with Nikki Marshall. So with that being said, and without further delays, I'm going to let the speakers each introduce themselves. So maybe we can start with Rashadi.
Thank you, Marisa Will. So my name is Rashadi Haynes II. I currently am at Bristol Myers Squibb. I'm a senior principal scientist, and I manage the immunomolecular toxicology group and co-manage the investigative toxicology group. I obtained my BS from Rutgers University, where I was pre-vet medicine. Then following that, I went to The Ohio State, did my PhD in retrovirology and immunology, and then postdoc at Sloan Kettering after that, where I really delved into getting my expertise in immunology. So I've been at Bristol Myers for approximately, 6.5 years now in this group. Time flies. And as I think about one of the major drivers that kind of brought me to this position and where I'm at in the field was I've always been interested in drug development. When I followed my postdoc, I knew I was going to go into industry. Following that, I went to a small biotech company where we worked on biosimilars, and I thought that was a great experience. However, I still wanted to get into a closer role into drug development from there. So then there was a position in the immunotoxicology group here at BMS, and I was really interested in it because it kind of mirrored a lot of the things I had been kind of training from graduate school and postdoc to then was developing molecular strategies to address problems with the immune system and developing protocols and assessments to evaluate immune parameters for different disease models or cancer models and such. So that was one of the big drivers why I wanted to be in this space, particularly in the non-clinical group here. So yeah, that's about it.
Thank you, Rashadi. Denise, would you like to go next?
Sure, I'll go next. Hi, everyone. My name is Denise Wilkins, and I'm currently the Senior Director for the Laboratory Sciences Group at the Charles River site in Shrewsbury, Massachusetts. I took kind of a circuitous trip here to where I am now. I didn't diversify much. I did all three of my degrees, bachelor's, master's, and PhD at the University of Nevada in Reno, Nevada. I originally started off on a pre-med track. I wanted to go and be a doctor. And honestly, my grades didn't end up where they needed to be after I'd finished my bachelor's degree. So I ended up looking for other options, and I got my first job in an immunology lab at the Portland VA Medical Center up in Portland, Oregon. And I learned flow cytometry, and I worked with animals, and I learned how to do injections and tumor models, and decided that was pretty much it for me. I had the opportunity to go back for my master's degree and work full time, and then ended up doing my PhD within that same lab where we focused on bone marrow transplantation, immunotherapies, again, a lot of animal work, a lot of immunology. I remember at the time I kind of lamented the fact that I had my projects were on a very macro scale. So it was working with animals. It was giving a tumor, watching the tumor shrink after therapy, but it wasn't really focused on, oh, what's this next molecular pathway, or how can I solve this one little question? Everything was on a macro scale. And at the time I lamented that, watching some of my other colleagues go through and present their posters and their dissertations. But I found that looking at things on that macro scale, especially with the immune system and the various components and how they interact, prepared me very, very well for the job, my first job out of my PhD, which was with Charles River Labs. So I started at the safety assessment site in Reno, Nevada. I was there for 7 years as a scientist, overseeing preclinical immunotoxicology studies, so flow cytometry, cell-based assays, functional assays. And it ended up, it was a great translation from all of those skills that I learned during my PhD into my first job. I had an opportunity to leave Charles River after 7 years, and I relocated my family out to the East Coast. And I worked in for Momenta Pharmaceuticals and preclinical development. That was a great opportunity to really kind of get a broader sense of the drug development process, working with analytical development teams and regulatory and CMC, and seeing all of the different parts and components that go into developing a drug. And then I worked at another small startup company, L-Star Therapeutics, before the big bad CRO sucked me back in, and I've been here at the Shrewsbury site for 5 years. My responsibilities here, in addition to overseeing some of the immunology and immunotoxicology studies, is also just to oversee a really diverse group of scientists and technicians. So any type of lab support, everything from bioanalytical to immunotox through to molecular biology falls under my purview. And I think I've talked enough. So I'll pass it back to the moderator. They can choose who goes next.
Great. Thanks, Denise. That's great. Should we introduce Shaji?
Yeah, hi. Good morning. This is Shaji Theodore. I'm a senior pharmacologist at the Center for Drug Evaluation and Research at FDA in Silver Spring, Maryland. I obtained my degree in veterinary medicine in India, practiced for 1 year, and decided I wanted to pursue research. I was basically very interested in pharmacology. So I did my master's in pharmacology in the same institute where I got my veterinary medicine. And then I went to a pharmaceutical company and worked for 1 year in the preclinical safety evaluation. Decided I needed more knowledge, so I came to US in 1999 to get a PhD in toxicology, and then spent several years doing research. So my goal was to have my own lab and do basic research, and my interests were in neuropharmacology, particularly Parkinson's disease and HIV dementia. So after several years, I just happened to join FDA. I wasn't planning to join FDA. I heard about FDA. I wasn't, I didn't know what was going on within FDA. I knew they regulate drugs. That's all I knew. So I've been here for like almost 8 years, and every year I have grown stronger in my belief in the mission of FDA. Basically, here what we do is to ensure that we get safe and effective drugs in the market, which is very important. The pharmaceutical companies are doing a very important job and service in bringing these life-saving drugs to the market. And I was a basic researcher. My thinking was all like, basic research thinking. The questions I was asking were all geared towards hypothesis-driven, investigation. So I had no idea how what we do in the FDA. So when I came here, I realized it was a very different world from what I was used to. The questions that we asked were very different from what I used to ask in the university. So here we always ask, what's the relevance of all these findings to a human? Whatever you see in vitro or in vivo with all these assays and tests, does it have any relevance to a human? And if it has any relevance, what's the benefit risk estimation here? Does the benefit override the risk? Ultimately, we have to keep in mind that people need medicines, and we just have to do our best to make sure that safe and effective medicines reach the market. So how do I involve myself in immunotoxicology? So we have several committees within FDA, genotox, immunotox, neurotox, all sort of things. And my research background was exploring adaptive immune response in Parkinson's disease. So there was an opportunity to co-chair the immunotoxicity committee within FDA. So I volunteered. And through that, I came to know about HESI and the work they do. So now I participate with HESI and try to learn from the latest developments in the field of immunotoxicity. And whatever I learn, I take them back to FDA and share it with my colleagues so we can enrich our knowledge. Thank you.
Great. I know people have such fascinating career paths. I love hearing about it. Okay, and last but not least, we have Marie.
Hi everyone, so I'm Marie. I'm a French Canadian, so I have a little bit of an accent. So my story, I'll start with school, and then I'll just take you to the kind of my career path. So I did biochem as an undergrad, and then right after that, I did some research, and we were working on, believe it or not, prototypes for CAR-T in one of my summer experience. So I kind of took that direction and went straight to PhD, which is not that frequent in Quebec because the expectation is usually do your master's first. And so I got started into a lab, and that lab was not a good fit for me. It was an immunology lab, and they work primarily, I would say, in immuno-oncology and also immunology in the context of HIV. But it was not a good fit because it was very, very, very crowded. And you'll see, it's a little bit the story of life, but I got married right after college, and I came back from the honeymoon with a little baby in tow, and I was not comfortable working in that lab. And so I ended up switching and doing a master's in neuroscience. It was funny because I went to the PI and I'm like, hey, hi, could I do my master's in your lab? By the way, I'm pregnant. But luckily, he just had a kid, and so he was super excited. So that went well, and I did my master's in neuroscience. But after that, I wanted to, pesticides were becoming more of a concern, and there was like a lot of kind of buzz in the news about health effects, and I was growing my family, and so I was curious about that. So I ended up doing a PhD on pesticides, and my PhD is technically in public health, but then I specialize in toxicology. So that's kind of school. But after that, the story of life continues, and we moved to the US. So I followed my husband a couple of times. We first went to Boston, then New Jersey, then California, then back in New Jersey. And so to that, I first finished my PhD from my basement in Boston. Then we moved to New Jersey, where I found a postdoc at Rutgers. And I thought I would stay in academia. But then, life happened and we ended up moving. So I ended up in California. And I was at the point where I needed like a real job, and so I just found this small consulting firm that had an opening. And so I kind of went in person, and that kind of really wowed them. And so they hired me. And I started working there. Then we moved back. And so when we moved back, I really also I didn't want to stay out of work too long. So I went into consumer product, but that was not quite my thing. It can be super interesting. I know people love it dearly, but for me, I wanted a bit more toxicity, I guess. And so I ended up did some more consulting, and then made my way into pharma after a couple of kind of little job move. And so I've been at Jazz for 6 years, almost. It's going to be 6 years in April. I'm a director of toxicology there. We at Jazz, the way we're structured is we do end-to-end. So we look from target identification through the development of the product. We usually follow our products to their life cycle. And so that means I would do, look at after the discovery program, do preliminary talks, then do GLP talks, and then do any additional talks that's needed dependent on the indication and the type of molecule. I work on biologics and small molecule. I work in neuroscience and hemato-oncology. And that's where kind of the immunotox comes in, because when you work in hemato-oncology, you often get immunotox. And I'm particularly interested in, so when it comes to immunology and immuno-safety, the current animal models they can tell you about some of the risk, but they can't predict the whole pictures because the immune system is so variable, right? And so it can, for example, in your animals, you could see an immune response like ADAs, but then in humans, it could be hypersensitivity or vice versa, right? So the specific of the response is not always exactly concordant between animals and humans. And that's where I think in vitro models can come in and provide us with additional information that's very interesting. And so I joined HESI, or I've been part of a committee per virtue of my affiliation at Rutgers. I mentioned earlier that I really loved academia and I really kind of left. So I teach undergrad and the GGPT, so the TOX program at Rutgers, where I teach actually risk assessment. And so I just recently contributed to a paper from HESI on first-in-human dose for immunomodulator. So that's about it for me. And I look forward to your questions.
Great. Thank you very much. And while I have you, Marie, I might ask you the first question, and then I might kick it over to Shaji after that. Oh, thanks, Charmaine. Perfect. Yeah, so, and you've already kind of touched on sort of that, thinking about those aspects of work-life balance, especially. So maybe tell us a little bit about what your kind of day-to-day looks like. So maybe give us a little bit more detail there, and how you handle that work-life balance. And you know, the third aspect is, what gets you up in the morning? Why do you love your job?
Yeah. And so I think that's the easiest to answer for me. So when any job, right, there's like the mission of the company, what you're doing, right? Then there's the people you work with. And so those are important aspects. Like, I love the people I was working with when I was in the consumer products industry, but the what I was doing was not kind of fulfilling. And so that kind of led me into getting involved with SOT or ACT and doing those, my kids, they call those the extracurriculars. So, but doing some of those activities, right, to kind of keep on publishing, keep my interest high into what was happening. And then right now, my job, I mean, I've put drugs in the market that saves lives. And so at the end of the day, that's very fulfilling, right? I'm proud of the work I've done. I'm proud of the findings I've had, and I know it's making a difference for the patients. So, what else can I ask for, right? So that's pretty fulfilling. And then in terms of the work-life balance, I think that is different for everyone, right? And it's even different depending on the stage of your life. So when I was in the consumer products industry for a little bit, it was, I'll say, more manageable to take care of the family. My husband was traveling a lot, so I was oftentimes I was joking and I would say like, yeah, I'm a single mom, but I cannot date, so I had to be home and go and pick them up at like 6:30 or 6 o'clock because, they charge you by the minute after that. And so I had to create that balance for me. And that balance is going to be different for everybody, right? And even if you don't have kids, if you have pets, if you have parents, if you have friends, like, you have to take care of them. So it's hard to kind of give specific advice. I would say I'm highly motivated. I like what I do, so I do work a lot. I definitely do more than the typical like 40 hours a week. But I do triathlon, so I also work out a lot, and I love cooking, taking care of my kids, but now they're older. So the baby is turning 18, so they're kind of off my day-to-day. They still ask me for stuff, but it's usually money. So
Sounds about right. Thank you. Okay. Yeah, Shaji, I don't know if you want to comment a little bit on this question as well.
Sure. A typical day, is spent looking at drug applications. It could be mostly it'll be investigative new drug applications when the sponsor, the pharmaceutical company or research investigators want to test this drug in humans. So they do non-clinical studies and submit it. And so I look at the non-clinical data and look at, any potential concerns for human safety, and whatever results I come up with during my review and my conclusions, I share with the medical team or quality team. We work as a team, so every day is spent reviewing, and a lot of times there are meetings. You have to go to meetings with the industry. Now it's virtual, and sometimes we'll do face-to-face. So this is to discuss with the industry representatives and sort out any issues. So I do occasionally review research grants. Also, we have working groups, so participate in working group once or twice a month. And you do have to write reviews, so not just look at non-clinical data and evaluate them, but you also have to write reviews to document. So a decent chunk of time is spent in that. Work-life balance is pretty good at FDA. They're very helpful. We share workloads. Like if there is in another division, if there is there are fewer people to review and they get more applications and they are swamped with applications, we do volunteer and share the workload. So and you can avail holidays. In fact, they encourage you take vacation and holidays, and they want you to be happy so you can come back to work fully energized. So work-life balance is pretty good, and I'm very happy. The rewarding part of my job will be, I'm doing my best to help bring new drugs to the market. So I'm associated with the drug when it's part of a new molecular entity, and that do all the preclinical studies, and it goes to the first phase, the phase 1, when you're putting the drug first in a human being, and then it moves through different phases, and ultimately it reaches the market. So I'm able to impact that development in a positive way throughout the phase, the development phase. And even after the drug is marketed, we do look at emerging safety data, and so we play a role there too. So that's very rewarding that I'm part of the I'm doing my best to ensure that we have safe and effective drugs. What gets me excited, it's my colleagues at FDA. I have pretty smart, motivated, and passionate colleagues, and we before coming to FDA spent a lot of years in academia. So we do have those interactions, like discussing scientific issues and questions and latest developments. So it's good to have pretty committed and passionate people around you, so you can bounce your ideas across. The troubleshooting, brainstorming sessions. These are very exciting and very rewarding.
Yeah, great colleagues, right? Really make a great work environment. All right, Marie-Soleil, should I pass over to you to do next question?
Yeah, thank you, Shaji and Marie, for your insightful comments and stories. So we're going to move to question 2. Denise, I might actually start with you. You did describe in your introduction that you had a non-linear career path, right?
Yes.
So I was interested in knowing, essentially, from your educational background, is there any specific experience that really prepared you for your current role right now? And as you were training as a PhD or postdoc, what were the skill sets that you developed that were the most transferable to the different positions that you've occupied throughout the years?
That's a great question. So the graduate lab that I came up through, in some ways, the graduate students were treated more like postdocs than actual grad students. So each of us had our own projects, but it was an immunotherapy lab. So we would have anywhere between 40 to 50 mice that were on a given study. They were tumor studies. We would have to give an immunotherapy and do takedowns. So it was too much work that any one individual could do at one time. So whenever one of us had a takedown, it was whoever was in charge of that would have to coordinate everything, make sure the documentation was ready, make sure all the reagents were prepped, but it was kind of an all hands on deck. So from that, you kind of learned leadership skills early on. You learned to be organized, because if you didn't do all of your homework and prep everything the day before to make sure everything was ready to go, the actual day of harvest was going to be a nightmare. It taught collaboration and teamwork. So how to delegate different parts of a project to other people. And then at the end of the day, it was kind of trust. And that's something that's, I think, as I've moved up and I've moved more out of the day-to-day science and more into management, delegation and trust is a big deal. Part of that comes down to just the quality of the relationships that you build with the people that work with you, and then your ability to kind of trust that you've invested in them to where you know that if you delegate a project or something, that they're going to be able to do it and do it well. I had the added benefit during grad school that, well, not benefit, the immunotherapy that we were working with in my lab was incredibly toxic. So while I didn't have any ideas about going into immunotoxicology when I was in graduate school, I was just studying immunology and thought immunotherapy was fascinating. Some of the histology that we took, I got a little bit of an introduction to some of the common endpoints that we look at and that we use in drug development and at a CRO. I had some exposure to that early on. So I've been able to kind of translate some of that knowledge and, oh, okay, this is targeting that cell population. This is in the gut. No wonder the guts were wrecked. And then kind of translate that in a CRO to working with, again, collaboratively with veterinarians. These are the clinical signs that you're seeing in the animals on a given study. To clinicians, okay, this is what's happening in these patients who are on these immunotherapies. These are some of the common side effects. So it's been gratifying to kind of see everything come full circle. So yeah, collaboration. And then I think the one other thing, and then I'll stop talking because I ramble. There's a certain level of customer service that's required from those of us that work at a CRO. So we contract with academia, biopharma, big pharma, everybody in between at a contract research organization. So it's not only customer service and how you deal and interact with the people who are your clients externally, but also your internal clients. So working collaboratively with you, Marie Soleil, my colleague up in Montreal, just really leveraging the network of individuals around you and working hard, not just kind of to your own ends, but just being a team player and working well with others, I think is definitely an important skill.
Thanks a lot, Denise. Rashadi, I'd like to hear your thoughts about this same question. You're in the industry, so your reality is slightly different. What are your thoughts about that?
Excuse me. Listening to Denise, it sounds like almost a mirror image of what I went through when I was in graduate school, where I came up in a virology lab, particularly retroviruses. I was working on HTLV1 and a rabbit model. And as a graduate student, you were responsible. I guess the big word I was thinking was responsibility, and learning that early on and being able to work with people cross-functionally as a graduate student, because I wasn't a veterinarian. I had to work with pathologists, veterinarians. I worked with people who are had more experience than me in different areas, and learning how to communicate with them when I was in early in my graduate career, to do necropsies on a rabbit that had a fully infectious molecular clone of HTLV1 in them. So that was kind of very daunting as a graduate student. So I had to learn all of those kind of soft skills of trust, being responsible, being organized, delegating, communicating with senior people and junior people within a graduate group. Also, my postdoc that kind of continued as I kind of got more into the molecular side of immunology, where I had to develop those skills again, and kind of in a more independent state where you're driving the project yourself and you're kind of responsible for every aspect of it. And not trying to be an expert in every arena and learning your limitations. And that was one of the things I learned. And it's kind of helped me as I transitioned into a pharmaceutical company here, where I'm at Bristol Myers Squibb. And back in the day in my postdoc, if I needed help with some type of RNA or PCR or something like that, you got to look it up, do it yourself, figure it out. Now it's an email. Now it's reaching out. Now it's collaborating with another group. And those are the things that you kind of learn to be able to synthesize information quickly. You don't have to be an expert in it, but understand overall what's going on and then trust the people you're working with. So those are kind of some of the similarities I saw between our experiences, Denise. And then I kind of was always interested, getting back to like the immunotox part of it, I was always interested in the safety aspect, because in certain communities that people grow up in, you always hear like, oh, you take this drug, it causes this, it causes that, it does this, it does that. And it's nice to kind of be in the arena where you can help explain this to some of the people in your lives, whether they be family and friends, kind of educate them too on how to interpret data and things that they see in the news and such. So that's kind of one of the things that I took from graduate school and my postdoc, is being able to synthesize information and communicate that. And I try to share that with students as well and people in the industry. So.
And that's an interesting comment, Rashadi. You were, I mean, in your graduate studies, it was really immunology oriented. Now you're kind of on the fence, right, of immunology and safety. Did you find that it was difficult to bridge both?
Yes, when I wouldn't say it was difficult. It was just a switch. When I'll say my biggest switch was probably the academic brain of like, okay, I get into safety. It's like, oh, all right, let's look at these subsets of T cells or regulatory cells, and you want to delve all into it. And they're like, no, that's way too much. And they're just like, all right, we're going to focus on these immune parameters. And I'm like, why not all the parameters? Why? And then it was kind of a switch in my brain saying, oh, okay, like these are the things that clinicians are interested in. These are the PD markers and biomarkers that they're going to take an interest in. These are things that are going to be able to translate to them actually running on a clinical trial. So once I kind of started getting an understanding of how it actually worked, then it was easier for me to transition early on, because, you know, as somebody coming out of discovery, you just want to get all of the information and answer everything, get all of the data. It's not always applicable to what you're doing on these GLP studies. So
you're very right about that. Thank you, Rashadi. Nikki, would you like to take question 3?
Sure. And Rashadi, while we have you, maybe I'll ask you this. So if you put yourself in the trainee's shoes, right, and you think about your career journey in immunotox, I don't know if you have any sort of stories to share or lessons learned, you know, that our listeners might benefit from hearing about.
Okay, I think one of the things that I kind of was always open to, I guess, was being open in the path of your career, because this wasn't necessarily where I thought I was going to be. I knew that I was interested in immunology and I was interested in safety, but I didn't actually know there was a connection until I learned about this position and got into the industry. So it was being open to what the knowledge I have can do for me in my career. So one of the things was that connected me actually getting this position was networking and having people that I'm connected to in the industry. And I always try to stress that to students and mentees that I work with, is the importance of networking with people when you go to meetings and when you're attending webinars and stuff, just the connectivity that you have with people in the industry, because that type of stuff will help you as you move forward in your career. And it will kind of, I think it helps connect you and gives you a better opportunity when the opportunity is there to take advantage of it. So that's kind of how I ended up in the position I am here, where I actually postdoc with one of the people who was in the group, and we were always friends and through on, and then they informed me about the position here. So it's kind of one of those things that I said there is an importance to kind of doing some of the time, some of the social stuff that you might not be interested in, or, you know, just connecting with people or talking to different people in different arenas. that I always kind of stress to people is very important for your career. So.
Yeah, totally agree. The opportunity to network at meetings on LinkedIn is so important, especially when you start doing the job searching.
Yes, and then secondly, I guess one thing to add to that too is a lot of people I find they're a lot kinder than you might be intimidated to reach out to people. And I always tell students, I think a lot of people in the industry are actually a little bit more helpful and nice than you actually, that LinkedIn page might seem very scary. I tell them, I might not get back to you in a week or 2 weeks, but I will get back to you. Reach out if you have any questions or things like that. So don't always be intimidated. I think a lot of people in the industry are always looking to help people who are really interested in the science and doing the work to come up. So.
Yeah, that's a great point. Thank you. Marie, do you want to comment on this question as well?
Yeah, sure, happy to. Anecdote or lesson learned. Well, I think I'll go back to just like the story of life that I was sharing earlier. You know, I think it's, if you love what you do, it's always easier to do it. And so I've always, you know, when I said earlier that I do work a lot, it's because I really enjoy it. Like I actually enjoy coming to my computer and looking at my data and doing all of that, right? So I think a lesson learned is try to find your fire, like, and sometimes, you know, you can be in school and maybe you just take like an elective because you're curious about it. I personally always loved immunology. I just thought it was just so fascinating because it's so complex and we're still learning about it, right? It's not a fixed science. And so I just, I don't know, I'm a lifelong learner. So I would say also don't be afraid to be curious, because ultimately, whether you go in immunotoxicology or immunology or something else, at the end of the day, you have to find something that kind of drives you to be motivated to get up in the morning and come and sit at your computer, even though I work from home, obviously, but I'll come to the bench, right? And the other thing too is probably the concept of delayed gratification. You know, sometimes when you're in grad school and you're working hard and you don't know where it's going, but I would say like if you keep working hard and you deliver, you'll end up, you know, you'll end up somewhere where you're actually happy and content with what you do. So sometimes it's worth to kind of push through the rough to get to the top of the mountain, right? Because it's hiking is just like that, right? You have to go to the pain of climbing to see the view. So I would say just keep at it.
Yeah, it's a great point. I always tell people, you know, when you're at that point in grad school where you're like, well, I can't do this anymore. Just if you get past that point, then, you know, the sky's the limit. As long as you keep, working hard, like you said. Okay. Marie Soleil, do you want to pass the baton?
We'll move on to question 4. So this is our last question for the panelists today. I just want to remind the audience, if you have any burning questions, please type them in the Q&A box, and we'll try to cover them in the next 9 minutes or so. Denise, quickly, and in 3 or 4 sentences, if you would have an advice to give someone that would like to pursue a career in the CRO business, what would it be?
Yeah, that's good. You gave me boundaries. I think you need to be strong scientifically, but also somebody who likes order in the chaos, because a lot of the work that we do is in a regulated environment. So it's not as simple at a CRO as I've got an idea, I'm going to go to the bench, I'm going to do it. There's a lot of documentation that comes into working in a regulated environment. So if you are a fan of organization, but also kind of willing to adapt to shifting priorities, then this would be a good job for you.
Thank you. And Shaji, at the FDA, what would be your advice?
Sure. For the FDA, I would several things. So as an academic researcher, you're interested in identifying a potential. You're discovering a potential for something, that can turn into, a useful drug. At the FDA, you decide what to do with that potential. Does that potential really translate into a reality? So basic science discoveries, when they are translated into some concrete products, right? Or your job at the FDA is to look at those data from basic science research and evaluate them and figure out, are they, usable? Can they be safe? So you are not just identifying a hazard, for example. You're not identifying an issue. You are characterizing that issue. If there is a problem in a in vitro or in vivo study, you should be looking at characterizing the problem, not just identifying it. Identifying a problem does not help anybody. You have to characterize it and use problem-solving skills, and also being open to advice from your colleagues and others, and always willing to learn and to look at that problem and see how it can be, useful. So you're looking for a solution, not just identifying a problem. So I would say that, keeping an open mind and looking at the problem and figuring out how you can move forward, and not just being satisfied with that problem being a roadblock. You have to help move that project forward. So I would say that being very open and flexible, because you're going to get a lot of input from the medical officers and people from product quality, related to chemistry, and clinical pharmacologists and statisticians. And there's a lot of brainstorming, and so you should be willing to change and be flexible and work as a team. It's not a individual thing, it's more as a teamwork, and everybody is interested in solving the problem and not being satisfied with just identifying what is wrong.
And
Thanks.
Shaji, from the previous positions that you've obtained, would you say that those skill sets are very specific to the FDA, or they're also applicable to the other positions that you've occupied prior to the FDA?
They were different. Most of what I was talking about was something that I learned after I came to FDA. So like a new employee, somebody who is coming from outside FDA first time, will have a period of time when they have to adjust to the thinking of the FDA. So in academia, like, I was more hypothesis driven. I was trying to identify some, issue and see how I can test that, by designing experiments, having specific aims. And here you have all the data in front of you. You have in vitro data, in vivo data, all sort of data in front of you. So you become good at quickly zeroing in on the relevant data that you have to look at, because you don't have much time. So you learn how to look at data and evaluate it pretty quick, and communicate your conclusions to the team and defend your conclusions, because it's not, it's not like I conclude something and that becomes a point that we convey to the industry. I conclude something, but I'll have to defend it, and to the team. And so teamwork and being flexible and putting yourself in the other person's shoe and looking at looking at an issue from a different perspective. Basically, it's being very open, so there's a lot of teamwork and being flexible and open. That's something that I was trained after I got into FDA. So if I'm looking for somebody, to hire within FDA, I want somebody who's, willing to learn, accept that, we don't know much. It's an ocean. In academia, that's an ocean. And within FDA, even now, I don't say that, I know a lot. I always, every day, I approach a meeting or my discussions with colleagues or even a review with the idea that I don't know much. I really have a lot to learn. And the best thing here is people are very willing to help, and you should not hesitate to reach out with questions. So I'm looking for somebody who is willing to learn, accept his or her limitations, and being flexible and being open for a change in the thought process. There's some people who are at the FDA for a long time, but who are not willing to adapt and learn, and who are very rigid in the thinking, and they don't get anywhere. Not much though. But people who are willing to learn and who are very adaptive, they can get quite far.
This is a very interesting comment, because sometimes the perception of people from the regulatory agency is that there's a certain rigidity associated to that. So the comment that you're making is very interesting, actually, that it's the opposite way, that you need to be flexible, actually, to be able to work in that environment.
Yeah. So it is driven by science. As long as I was outside FDA, I did not know how it worked. But within FDA, I realize every decision that we make, at least I can speak for my division, and my office. There are like 4 divisions within that office. We go only by science. Is it justified, by science, or is there a strong scientific underlying, principle that supports my decision? So it's still science. There are regulations, there are guidances. The guidances are there to guide you, and they recommend a certain course of action, but they are not set in stone. You're not obligated to follow exactly the guideline. So you ultimately rest on science and what science tells you. If you're satisfied, then if you're satisfied that the answer is based on science, then that's all matters.
Yeah. Thank you, Saji.
No problem.
With that being said, I think, Charmaine, I'm going to turn it back to you. We're almost at the top of the hour and a half, the hour here. So I would like to thank all of the panelists for answering the question, giving their perspective about their career. I really hope that this was useful for the students on the line. And as Rashadi mentioned, if you have any questions for anyone that was on the call here today, please reach out. It's going to be our pleasure to answer those.
Yes, I mean, again, huge thank you to all the panelists and the moderators involved. Speaking of oceans, I certainly learned a lot. And every time I actually engage with the scientists from the Immuno-Safety Technical Committee, I feel like I am learning oceans of knowledge. I'm learning something new every day. And that's what makes my job very exciting. But also, I get to interact with the creme de la creme. Like if you saw the titles here, there are directors and senior scientists and senior principal scientists here that I get to talk to and learn science from. So it's really exciting and amazing to me. So I'd like to thank you all for sharing your time and your wisdom and your advice with the students here today. And those who will review this, because this is recorded. So I'm sure this will be an excellent resource from any student who's interested in maybe meandering through the path of immunosafety sciences. So with that, I would also like to mention that Dr. Denise Wilkins not only a wonderful science at Charles Rivers Laboratory, but she's also stands as the science outreach lead for the working group that helped to organize and bring forth this particular event. So I wonder if you have any closing remarks to the students before we close. I know that there'll be a survey afterwards, but any last words of wisdom that you'd like to impart to the team?
Well, first, I'd like to thank you, Charmaine, for the pivotal role that you play in bringing all of us together and providing this as an awesome resource. I also want to thank the students for taking the time to call in and just kind of learn. Hopefully, you've been able to learn from our experiences. Scientific outreach is one of the key proponents of the ITC and what we do here. So I hope you've gained a lot out of it. As Charmaine mentioned, there's a survey at the end. Please take your time to fill it out. Let us know, good, bad, ugly, your feedback on this. We want to continue to provide meaningful opportunities to engage with individuals throughout all parts of their career, from the beginning to mid-career. We will be having another career webinar later this year, focused on mid-career individuals. So yeah, thank you very much for your time and for your attention. And again, thank you, Charmaine, for all that you do for us and for the other speakers, and to our moderators, Nikki and Marie Soleil.
Thanks so much, everybody. Enjoy the rest of your spring break.
Thank you.
Bye, everyone.
Bye. Have a good day.